Hi, so I'm sitting here now talking with Rafaela Gisuete from DTxPharma. DTx is one of our partners working on a CMT1A program. And we're here to talk a little bit about the program that they're doing at DTx, why they got involved with CMT, and what they've learned about CMT since working with us. So hi, Rafaela.
Hi, how are you doing?
Well, thank you. Thanks for joining me today. So I know that you're the lead scientist on the CMT program, Rafaela, and I'm so excited to have you as a part of the team. And you have your PhD in neuroscience, right?
Yes, I got my PhD in neuroscience in 2010 in Milan, Italy. And after that, I moved to Portland, Oregon, in the US for my postdoc. So I worked a little bit with Alzheimer's disease and then with brain stroke for a lot of time.
Great. And how did you get into CMT? Talk a little bit about that.
Sure. So I joined DTx Pharma about 7 months ago. And, I joined and I really wanted this job because I really think that RNA therapeutics are the next generation of drug that they are going to be able to cure or treat genetically induced disease. So, and as soon as I started here, I knew about this project about CMT 1A, and I have a neuroscience background, so I thought that would be the perfect fit for me. So I could put together two things that I really like. So my love for neuroscience together with my belief that the RNA therapeutics can be the next generation of drugs.
So can you talk a little bit about the RNA therapeutics and what you all are working on at DTx and why it applies to CMT?
Yeah, sure. So we are particularly interested in CMT1A because we know that it's a genetically defined disease. So we know that is due to the overexpression of the PMP22 gene. So the natural way I would say that you can think about how to treat or cure this disease is to decrease the overexpression of PMP22. So, and the RNA therapeutic is just perfect for that. So what RNA therapeutic means is that we can use double-stranded RNA-mediated interference to decrease the product of the expression of this PMP22. Now, of course, the biggest challenge with the RNA therapeutics is that is drug delivery.
Mm-hmm.
So cells are not taking up easily the RNA, the small interference RNA. So here at DTx, we have a technology where we combine the siRNA with the fatty acids. And all our cells express receptors for fatty acids. So the idea behind that is that combining this fatty acid with the siRNA, we can increase the delivery of our compound inside the cells. And once the compound is inside the cells, can target this gene. So basically, it can combine with the RNA of the gene, and that is going to lead to the degradation.
So if I understand it correctly, I have CMT1A. And so most people who don't have CMT1A have two copies of this PMP22 gene. And lucky me, I got the third copy, and I inherited that from my mom. And what I understand your technology can do is really decrease the expression of that gene and therefore decreasing the expression of the protein. Is that fair? And really what we want to do is take it from 2 down, or from 3 down to 2. Is that fair? Is that an overgeneralization?
No, that's perfectly description of what we are trying to do. In particular, we are CMT1A is particularly interested for us because we only need a 30% of reduction of the PMP22 to get to the, to see a therapeutic benefit. Otherwise, actually, you know really well, right? The PMP22, if it's expressed too much, leads to demyelination of the peripheral nerve. But if it is too low, it's also a problem. So there is a fine balance that you need to get of PMP22. So in this case, this is a perfect proof of concept for us, for our technology, because you only need a 30% modest reduction of your gene expression. The biggest challenge for this field, for the RNA therapeutics, is delivery. Mm-hmm. And, as I told you before, all our cells have these receptors for this fatty acid that we are going to attach to the siRNA. So I think that the new thing about our technology is that potentially you can go everywhere. And, we are also working on our technology to get a binding to the albumin that is in our blood, and it goes everywhere. So we can think about the albumin like a bus, and our compound can bind this bus so it can go around, so increase the time of exposure of all our cells to the compound. The siRNA that we design are specific for PMP22 in this case. So they're going to target only the cells where PMP22 is increased.
So, it's really exciting that we found this match, that your technology, especially using the fatty acids and the peripheral nervous system and the siRNA, kind of all mixed together to get into the peripheral nervous system for CMT. It's awesome that we found this technology and that we found this partnership. Can you talk a little bit about how DTx and you came to CMT and where the interest is and how that all started?
So yeah, we are DTx. We didn't know anything about CMT1A before meeting you. So our CEO actually met you at a conference. You guys started talking. And again, we thought that it was a perfect proof of concept to test our technology. And so everything started there. So the CMT Research Foundation gave us some money. We had a little bit of money from you. Thanks, to that little funds, we were able to generate some first preliminary results, and then we used those data to write a grant. And now we have an SBIR grant that we are currently generating that data for that. But I mean, our interest in CMT1A doesn't stop there. So our idea is to generate more data, and with the other data we can find some other partnership with big pharma that are interested in that, and we can generate interest in some other investors, so we can raise together more money to fund these studies.
Our small investment created enough data to get NIH funding, which is where the SBIR comes from the National Institutes of Health. So now we have government dollars involved in this project. And you all have invested some of your own money from DTx, which you wouldn't have done otherwise if the data weren't compelling. And now you're talking to larger investors and even big pharmaceutical companies about carrying forward this idea. And so a little bit of money from the patients can turn right now. I mean, we're looking at sevenfold our invested already.
It's it takes a village.
Right.
So it's really exciting for us as well, because I mean, again, from nothing, we didn't know anything about CMT1A, and now we have a project going, and we are really excited to keep going, because we are going to get there. In the project I'm working on, we are designing a lot of siRNA. We are conjugated them, and the first thing that we are doing with these conjugates is to test them on cells.
Mm-hmm.
So we are testing them on cells. We After we get the data, we select one that are more potent in reducing the PMP22 expression, and then we are going to test them in vivo target engagement. That basically means injected this compound in mice that have the disease. So, and I think we are at this point right now. So we are gonna test soon, in the next month, I think, our compounds in mice that express the phenotype of the CMT1A, and that would be actually the very first proof of concept that our technology works, hopefully. And from there, new things are going to happen, because we can twinkle a little bit the compounds to make them more potent. We can test new motifs of our fatty acids, and then of course we are going to do dose response and efficacy study. Of course, it's a long way. It's going to take a lot of time, a lot of work, and a lot of people. But as I was telling you before, it takes a village, and we are all committed to get there.
We're committed to helping you understand everything there is to know about CMT, to help you design your experiments better. So I'm going to walk through this process a little bit. First is testing it in the cells that have the CMT1A, that overexpression of the PMP22. Then the second phase is testing it in the animals, these mice that have CMT1A. And instead of having 3 copies of the PMP22 like I do, my understanding is that this mouse has actually 4 copies, and that you're trying to decrease it down to the 2, which is what we call the wild type, or the one that doesn't have the CMP. Is that right?
Yes. So we are going to test our compound on these mice that actually express the human gene of PMP22. So yeah, the idea is to do first target engagement. That means we can show that we can decrease the expression in these mice.
Mm-hmm.
And then the second step will be a dose response. So we can test different
Uh-huh.
doses of our compound, and then there will be the therapeutic, the efficacy study, where we can test, for how long, or when we need to treat the animal, and for how long, when we can see the slowdown of the disease.
Right.
And of course, there will be a safety study to make sure that our drug is a safe drug, and that there are not going to be side effects.
And how long typically does that kind of process take? I mean, I know in 4 to 6 weeks, we're hoping to test the animals and see how it works then. And then for some of those other stages, for the safety and the efficacy and the right dosage, like what is typically a timeline that one could expect?
Uh, let's see. Usually, therapeutic efficacy study, they're done in 12 weeks.
Okay.
But that will be after the dose response and everything. So it's gonna take, I would say, at least 1 year to
Okay.
get some nice data, that then we can push forward.
Having a partner like you and DTx to advance this, and your enthusiasm, your investment, your commitment, your weekend work and evening work for us. Thank you. I don't know how else to say it. Thank you.
Thank you for your help. I mean, we are here because of you.
Well, it's an awesome partnership, and we commit to keeping the public informed about the process of this project. And when we can share the results, we will, whether they're positive or negative, because I think we owe that commitment to our community to show them this is what we've learned. If it doesn't result in a drug, this is what we learned from it and how we can move that forward as well. So every investment is a worthwhile investment, even from the things that we could learn to do differently next time.
Yeah, yeah, definitely. I, I've already said that, but it's a long way. There will be ups and downs,
Mhm.
uh, but we are committed to get there.